Hello, everyone. My name is Selin, and today I will be talking about an initial phantom study for an office-based low-field MRI system developed by Promaxo Inc. The objective of the study I will be presenting on today is an initial assessment of the navigation accuracy of the Promaxo MRI system for prostate interventions. I would like to start off by briefly talking about the need area, prostate cancer. Prostate cancer is the second most prevalent cancer in U.S. men, with 1 in 7 men being diagnosed during their lifetime. It is also the second leading cause of cancer deaths in American men. For stages 1 through 3 prostate cancer, that is, cancer that is still local or regional, the 5-year survival rate is almost 100%. However, for stage 4 prostate cancer that has spread to other parts of the body, the 5-year survival rate drops to 30%. Early diagnosis could be crucial in saving a patient's life. There are different approaches to prostate cancer diagnosis. The most common approach in the United States is an ultrasound-guided biopsy. An ultrasound probe is inserted into the patient's rectum to localize the prostate, and samples of the prostate tissue are taken in a systematic manner. Because ultrasound is not specific for cancerous lesions, and the systematic approach samples a limited region of the prostate, cancerous lesions can often be missed. Another approach is MRI ultrasound fusion, in which a previously acquired MRI image with identified suspected lesions is overlaid with live ultrasound images. However, the ultrasound transducer has been shown to cause prostate gland deformation, which could lead to large error in targeting. Finally, prostate biopsy directly under a standard MRI is cumbersome and cost prohibitive, as it requires special infrastructure within hospitals. The Promaxo MRI system is an open, single-sided 0.066 Tesla MRI machine. The system consists of a magnet card and an electronics rack, as shown in the image on the left. The rectangular electronics rack houses the electrical components of the system. The magnet cart houses the magnet, gradient coils, and transmission coil, and attaches to the rigid external pelvic receive coil, which is worn by the patient during the procedure. For prostate cancer imaging, the patient is in a lithotomy position with their legs around the magnet cart, as shown on the image on the right. The imaging field of view is 6 inches from the face of the magnet, has a diameter of 4 inches. The system has recently received FDA clearance for prostate biopsy navigation. Given its novel magnet form factor and small size, it can be placed in a physician's office. The system uses pre-programmed pulse sequences, making it easier for a physician to operate. Due to its field inhomogeneity, the system uses nonlinear reconstruction algorithms instead of the traditional Fourier transforms. The variably tuned receive coil allows the system to acquire multiple slices simultaneously. In the proposed clinical workflow for prostate biopsy navigation, a T2 scan of the region of interest will be taken. The external receive coil has a template grid and a fiducial holder attached to it, which embeds external fiducial markers visible in MR images. The fiducial markers are used in order to localize the template grid relative to the prostate. After obtaining the image, the urologist will localize the external fiducial markers on the graphical user interface, as shown in the middle image. Once targets are identified, needles are inserted into the grid location and depth calculated by the UI, and a sample can be taken. The image on the left shows slices of a T2 scan of a prostate phantom, and the image on the right shows a prostate phantom box with a template and a template holder attached to it. This initial study to assess the navigation accuracy of the low-field Promaxo MRI system was performed on prostate phantoms. 5 phantom sizes were used to span a range of prostate volumes and different prostate shapes, as can be seen in the image on the left. Prostates from different patients were segmented to generate a 3D model, and molds in the second image were created from them. Materials for the phantom were experimentally determined to mimic parameters for human tissue. First, a standard T2 scan of the phantom and fiducial markers was obtained on the low-field Promaxo MRI system. 3 or more needles were then inserted into each phantom, and the coordinates and depth were recorded. Next, a 1.5 Tesla T2 scan of the phantom was taken. The 1.5 Tesla was used as ground truth in the study. To obtain the ground truth coordinates, 2 urologists identified the needle track on the 1.5 Tesla image. In order to compare the planned target from the low field scan and the ground truth, the template was localized by 4 urologists, and the low field and the 1.5 Tesla scans were registered or overlaid to be in the same frame of reference. Registration was performed using the boundaries of the phantom box. The area was calculated in 2 different ways. The first way was the center-to-center distance between the planned target and the ground truth target, shown as a blue line on the diagram. The second way was the shortest perpendicular distance between the planned target and the entire core segment, shown in black on the diagram. This area measurement is acceptable because the biopsy will be taken somewhere along within the biopsy core. 5 millimeters was taken as the accuracy limit, as clinically significant cancer is defined as having a volume of greater than or equal to 0.5 cubic centimeters. The radius of a spherical tumor of this volume is about 5 millimeters. For the ground truth measurements, the average distance between selected points across the 2 users was 2.32 millimeters with a standard deviation of 1.51 millimeters. The average center-to-center distance was calculated to be 2.57 millimeters with a standard deviation of 1.02 millimeters, and the average center-to-line distance was 2.05 millimeters with a standard deviation of 1.24 millimeters. Both of these were well below the 5 millimeter accuracy limit. Inter-observer variance between 4 urologists was 0.0029 and 0.039 square millimeters, respectively. The results of this initial study are promising. The system is currently being used to perform biopsies on patients in clinic. Future work on navigation accuracies should include a larger number of urologists and an error analysis to assess the contribution of different types of error to the overall error. The system should also be continued to be used on patients in clinic. In addition, the navigation accuracy of the system should be compared directly to MRI ultrasound fusion navigation accuracy. Thank you for listening.